Hi viewers, in this class we are going to discuss the neutralization apart from the antigen antibody reactions. Under this topic, we will discuss the introduction to the topic, mechanism of neutralization, types of neutralization and common neutralization tests. In general, antibody antigen antibody reaction that can block the harmful effects of the toxins or the viruses is the neutralization. Neutralizing substance is an antibody produced by the host, which is known as the antitoxin. Antitoxin combines with the toxin and the toxin get neutralized. Toxin antitoxin neutralization tests. In neutralization reactions, the harmful effects of a bacterial toxin or virus are eliminated by a specific antibody which is commonly known as a antitoxin. An antitoxin is an antibody produced in response to a bacterial toxin or virus that neutralizes the bacterial toxin or virus and therefore no harmful effects are produced. Neutralization of a virus is defined as the loss of infectivity through reaction of the virus with specific antibody. Virus and serum are mixed under the appropriate condition and then inoculated into cell culture, eggs or the animals. The presence of unneutralized virus may be detected by reactions such as CPE, heme adsorption and hemagglutination, plaque formation, and as well as a disease in animals. The loss of infectivity is brought about by interference by the bound antibody with any one of the steps leading to the release of the viral genome into the host cell. Here we are observing the toxin and antitoxin neutralization. Here it is a positive test and here it is a negative test. Here we are observing the toxin, that is hemolysin, and serum which contains the antibody and RBC lysed with the toxin. Testing the presence of the antitoxin in the serum. If they are present in the toxin will be neutralized and RBC not lysed. If there is no antitoxin, the toxin will lyse the RBC. So that is a toxin and antitoxin neutralization. Here there is a neutralization test, RBC, antiviral antibodies for serum, viruses. Here viruses get neutralized and hemagglutination is inhibited. This is the neutralization of the toxin by the antitoxin. There are two types of neutralization, namely reversible neutralization and the stable neutralization. Let us see one by one. Reversible neutralization. The neutralization process can be reversed by diluting the antigen antibody mixture within a short time of the formation of the antigen antibody complex, that is usually within 30 minutes. It is thought that the reversible neutralization is due to the interference with attachment of virions to the cellular receptors. Example, the attachment of hemagglutinin protein of influenza viruses to sialic acid. The process requires the saturation of the surface of the virus with antibodies, whereas see the stable neutralization. With time, antigen antibody complexes usually become more stable after the several hours of the antigen antibody reaction, and the process cannot be reversed by dilution. Neither the virions or the antibodies permanently changed in stable neutralization. For the unchanged components can be recovered. The neutralized virus can be reactivated by proteolytic cleavage. Stable neutralization has a different mechanism to that of reversible neutralization. It had been shown that neutralized virus can attach and that already attached virions can be neutralized. The number of antibody molecules required for stable neutralization is considerably smaller than that of reversible neutralization. Kinetic evidence shows Kinetic evidence shows that even a single antibody molecule can neutralize a virion. Such neutralization is generally produced by antibody molecules that establish contact with two antigenic sites on different monomers of a virion, greatly increasing the stability of the complexes. An example of stable neutralization is the neutralization of polioviruses, whereby the attachment of the antibody to the viral capsid stabilizes the capsid and inhibits the uncoating and release of viral nucleic acids. Here, the neutralization test. Bacterial exotoxins are capable of producing neutralizing antibodies, antitoxins, which play a protective role in diseases such as diphtheria and tetanus. Here, there is a toxin antitoxin neutralization can be measured in vivo and as well as the in vitro methods. Toxin neutralization tests. Bacterial exotoxins, they induce the neutralizing antibodies which are called as the antitoxins, protection against diseases such as diphtheria and tetanus. And the in vivo tests of neutralization, they are namely toxigenicity test, detection of toxin of Corynebacterium diphtheriae, Schick test, and as well as in vitro tests which are conducted in laboratory glassware, anti-streptolysin O test, ASO test, virus neutralization test, nasal reaction or rapid detection of C. welchii. Here there is a detection of toxigenic strains, Elek test. It detects the toxicity of Corynebacterium diphtheria, which is extremely important in the diagnosis of diphtheria. Filter paper impregnated with diphtheria antitoxin is laid crosswise to the organism. If organism produces the toxin, then a precipitate will form at 45 degrees angle from the filter paper. If toxin is not produced, then no precipitate line forms. This is the Elek test to detect the toxinic strains. And coming to the Schick test, it is used to determine the susceptibility of a person to diphtheria, named after its inventor Bela Schick in between 1877 and 1967. The Schick test is no longer in use. The availability of safe and effective toxide preparation has made susceptibility tests unnecessary. And Schick test procedure: a small amount 0.1 ml of diluted diphtheria toxin is injected intradermally into the forearm of the person, and you see the minimum lethal dose MLD as LD minimum is the least amount of drug that can produce death in a given animal species under the controlled conditions. Similar dose of inactivated toxin, which is heated at 70 degrees Celsius for 30 minutes, is injected intradermal into the other arm as a control. First one is the practical one, test sample. Second one is the control. And the readings are taken after the 1, 4 and 7th days. And how to interpret the results of the Schick test? Results can be interpreted as positive reaction, where when the test results in a red necrotic area 5 to 10 mm diameter within 24 hours, reaching a maximum within 4 to 7 days, positive test indicates susceptibility to diphtheria and thus requires immunization. Negative reaction, where no erythema, no any on any arm, indicating that the toxin has been utilized by the antitoxin and the person is immune to diphtheria. And some of the times there is a pseudo positive case when there is only a red colored inflammation within 6 to 24 hours and it disappears rapidly within 4 days. This reaction same on both arms and indicates immunity. This is the interpretation of the results of Schick test. And antistreptolysin O titer or ASO. Antistreptolysin O or ASO. Here there is a streptolysin O is the antigen. Antistreptolysin O is the antibody. For the indicator system we have used RBC. So here there are the serial dilution method. 1:4, 1:8, 16, 32, 64, double dilution. And right from left side to right side, there is a hemolysis where we will find out the end point. Here there is a positive titer greater than 200 units for the ml. It detects the antibody to the antigen streptolysin O produced by group A streptococci. Titer rises to a peak at 4 to 6 weeks and will remain elevated for 1 year. Positive in streptococcal infection, example upper airway infections, scarlet fever. Post streptococcal infection complications, example glomerulonephritis and rheumatic fever. And false positive in some bacterial infections. Here, this is the antistreptolysin O. It is positive test indicator. It is negative and it is also negative. Antistreptolysin O negative. And neutralization test, virus neutralization test. Neutralization of viruses by their antibodies and are demonstrated in cell cultures, eggs, and as well as animals. Neutralization of bacteriophages by the plaque inhibition test. Plaque inhibition test. This is the nasal reaction. Here, this is the procedure, and this is the positive result of the nasal. Nasal reaction is a biochemical test that is used to identify organisms which liberate phospholipases or lecithinases, example Clostridium perfringens. The alpha toxin of Clostridium perfringens has phospholipase activity, and hence when grown on a medium containing egg yolk phospholipid, the organism can break down this insoluble triglyceride. Phosphorylcholine release is seen as an area of opacity around the bacterial colonies. Antitoxin can inhibit a positive response. So by this we conclude the neutralization test apart from the antigen antibody reactions. Thank you one and all.